This presentation will provide an overview of essential techniques for insect collection, preservation, and identification. We will discuss the importance of collecting insects for research and education, as well as the proper methods for pinning, mounting, and identifying various insect species. By mastering these techniques, we can contribute to our understanding of the fascinating world of insects and their role in ecosystems.
Before anything else, how can we observe insects without an insect specimen? This is where insect collection comes in. Insect collection is the process of gathering, preserving, and organizing insects for scientific study, research, or educational purposes. Its ultimate goal is dependent on the observer's question. Some may collect insects as a hobby, and some for practical utilizations in different fields, such as biodiversity studies, identification, pest control, public health, etc. Such practical uses are the reason why the collection of insects is relevant, because it contributes to research and practical applications in various fields. In our second laboratory exercise, the ultimate goal of the students was to collect insects to be utilized for pinning and proper identification. The procedures included an active collection, which was through general hand collection and with the use of sweep net, and a passive collection, which was through the utilization of baits. In the end, both procedures succeeded because the students were able to collect insects.
The method of pinning insects and paper stage pinning are essential techniques in entomology. It is employed to preserve and display insect specimens for scientific study. The paper stage pinning is often used for delicate or smaller specimens. In this case, we utilize an ant, securing them in place without damaging their fragile bodies. The materials used for paper stage pinning included entomological pins, paper tips, a probe, adhesive, a Gucci ball, and of course the ant specimen. A small tip was placed on a pin using a pinning block to ensure the correct height. This pin was placed on an Iguchi ball, then a small amount of adhesive was applied to the end of the paper tip. The ant was positioned with the probe placed just slightly to the right of its midline, specifically through the second segment of the thorax, ensuring the body was securely attached to the paper tip without damaging key structures. This method preserved the specimen's integrity and prevented damage, ensuring it was suitable for long-term study. Paper stage pinning provided an effective method for preserving delicate insect specimens, like ants, by securing them in the correct position without causing damage. This technique ensured long term preservation while allowing for clear visibility and study of the specimen's anatomical features.
Following the pinning process is mounting. It is a basic method used in entomology to mount insects for examination. It provides a stable way to preserve and observe their physical structures, making it easier to study their features for identification and research. The materials include an Iguchi ball with an entomological pin and paper tip, water-based glue, an entomological probe, and data and determination labels. A small amount of white glue is applied to the paper tip to prevent excess coverage. The ant is placed on the paper tip under a dissecting microscope with its dorsal side up and the left side positioned so that the paper tip is in between the first and second pairs of legs. After examination, the specimen is labeled. In summary, mounting through pinning is crucial for preserving and examining ants in entomology. Proper orientation, minimal glue use, and accurate labeling ensure that key features are visible and the specimen is ready for study and identification. Following this, the ant identification will be discussed by Ms. Christine Arcilia.
Taxonomic identification of a species provide an information on the biodiversity of an area and aid in developing a control measure if the specimen collected were a vector of a certain pathogen. To properly identify a species, aside from molecular techniques, researchers use dichotomous key. Dichotomous key is composed of various couplets that has two different statements which describe the morphological characteristic of a species. In those two statements, you need to identify or to choose the appropriate description that perfectly illustrate the collected specimen. Afterwards, if the specimen were mounted, the taxonomical identity of the species should be placed in a determination label and pinned along with the identification label and the paper stage. In a previous laboratory activity, we need to identify the ant specimen that were mounted. And in order to do that, we utilize the identification keys from the study or from the Journal of Zookeys.
So, the examination of mosquitoes, particularly Culex and Anopheles species, is significant. So, these mosquitoes are vectors for diseases like malaria, which is transmitted by Anopheles mosquitoes, and filariasis, as well as the West Nile virus and encephalitis, which is spread by the Culex mosquitoes. So, in identifying and differentiating these mosquito species, it helps in understanding the epidemiology of these diseases and helps in terms of implementing effective control measures. So, the mosquitoes that belong to the family Culicidae are more notorious vectors of several diseases. Among the many genera of mosquitoes, Anopheles and Culex species are important due to their roles in transmission of malaria and other tropical diseases. Also, the specific features of mosquitoes' anatomy, such as their head, thorax, wings, and legs, are examined. Also, based on the mosquito we've examined, it is an Anopheles, and the difference between the two. So, for identifying the Culex mosquitoes, the head, especially the palps in the females, its proboscis is shorter, and its thorax is covered with plain scales. It is also often brown. Its wings are not spotted, and the scales of its wings are uniformly brown, while its resting position, so the body remains parallel to the surface on which they rest. And as for the Anopheles, so if you can see its head in females, the palps are as long as the proboscis. Its thorax is usually covered with black or dark brown scales, or sometimes it also has a distinct pattern. Its wings is characterized by spotted wings or light scales. We've also examined its halteres and flight wings. Lastly, its resting position, so the body rests at an angle to the surface with the abdomen, so its abdomen is raised. And so overall, the identification of mosquito species, particularly the Culex and Anopheles, plays a critical role in managing mosquito-borne diseases. So by recognizing its distinctive morphology as well as understanding its behavior, it is essential, especially in medical entomology, to effectively identify the disease vector.
In entomology, collection, pinning, mounting, and examination techniques are crucial for effective insect taxonomy and research. Proper collection methods, such as sweep netting and light traps, ensure a diverse and representative sample. Once collected, insects are typically pinned to preserve their morphology, which is vital for accurate identification. Mounting techniques, whether on slides or in jars, allow for detailed examination of anatomical features under magnification.
For our synthesis, we have observed that these methods not only facilitate species identification, but also contribute to understanding ecological roles and evolutionary relationships. The systematic approach of these techniques ultimately enhances the reliability of taxonomic classifications. As such, mastering these methods is essential for both amateur and professional entomologists. In summary, effective insect handling techniques underpin significant advancements in entomological research and conservation efforts.